It is my pleasure to welcome you to the 10th annual Nathan S. Greenfield Family Lecture in Pharmacology. This lectureship, sponsored by the Greenfield family, honors the patriarch of the family, Nathan Greenfield, his wife, Corinne Sternheimer Greenfield, their only child, Dr. Rosalie Greenfield Weiss, and her husband, Dr. Raymond Weiss, and their daughter, Lynn Stewart Weiss. The family roots began here in Cleveland, where Nathan, a pharmacist, operated the Wade Park Pharmacy until 1956. His wife, Corinne, was a concert pianist who studied at the New York School of Music, which is now known as the Juilliard. Their daughter, Rosalie, was born in Cleveland and combined the talents of her parents, first as a ballet artist and then as a pre-med student at the Mather College of Western Reserve University. Her study was interrupted by service in the FBI during World War II. Then she completed a degree in psychology and eventually formed a practice with her husband, Raymond, who was also a psychologist. Their multi-talented daughter, Lynn, tragically died of cancer in 1971, having completed all of the requirements for the Ph.D. degree in public law and government at Columbia University, except for the dissertation. We are very appreciative of the family for their generous sponsorship of this lectureship, as well as a travel award each year for a graduate student who has displayed exceptional skills in presentation. I now ask Chris Steelvis to introduce our honored speaker.
It's a real pleasure to introduce today's speaker, Dr. Wayne Hendrickson from the Columbia University. He's a Harvard Hughes investigator there. Wayne is one of the leading structural biologists in the world, and his contributions to biology is multifaceted. He's a tremendous methods developer, and he's developed a technique called multiple anomalous dispersion, or MAD, which is widely used in the post-genome era for determining de novo structures. And in fact, most of the structures solved recently probably have been solved using this method. He's also made tremendous contributions in the structures that he has solved to elucidate very important mechanisms in biology. Some of these structures, I'm not going to name all of them, but a few of them, the GP120 and the CD4 complexes that are involved in HIV propagation. His works on chaperones that are involved in protein folding, and also some viral reverse transcriptases, as well as several other structures. He's the recipient of several important awards, and I think I counted about 20 of them, so I'll just mention a few of them. He won the Patterson Award for crystallography, the Fritz Lippmann Award, the NIH Merit Award, and then here's an interesting one, Mayor's Award for his contributions in science and technology from the city of New York. He's a member of the National Academy of Science and is, of course, a Harvard Hughes investigator. Wayne is the editor of journals such as Current Opinions in Structural Biology, and also I think he was the previous editor of the journal Structure. He is the chairman of the Structural Genomics Consortium. Wayne's interests in science include a lot of structure-function studies involving membrane receptors, as well as cellular signaling, molecular chaperones and protein folding, and viral proteins and HIV infection, and is involved in developing techniques in structural biology. So I would like to invite Wayne, and it's a real honor to have him here to come and present his talk.
And to remind you of this honor and occasion, we have a commemorative plaque.
It's beautiful.
For you to cherish.
Thank you very much. I'll remember who I am while I talk, I guess. Well, thank you, Chris, for that very kind introduction, and thanks to the Greenfield family for the opportunity to be here in Cleveland again and to talk to you about some of our work. Now, I'm probably a little off base here. I'm going to give you a talk that in some ways is a review. I'll have some new things to tell you about, but I wanted to be a little bit comprehensive on this occasion with the title that I've given you, which is to address the kind of question of, I think, quite central interest for biochemistry and biophysics, a mechanistic question of how signals actually manage to go from the outside of a cell to the inside of the cell. Signaling is a big deal. We know a lot about signaling. We know there are a lot of things that happen inside the cell. But there is, I think, an interesting mechanistic question about how these diverse molecules actually respond to many different kinds of ligands, the problem of pharmacology, if you will, in doing this. And there are molecules, I'll mention something about G protein-coupled receptors that are important. Acetylcholine receptors are cis loop receptors. I'll say something about those. I'm not going to be talking about some of these other kinds of molecules, but all of them face the same, generically the same kind of problem. And the sense I want to leave you with is that there is no single solution, probably not even a single solution for any one of these classes, but they each present a certain particular problem. So for example, we have the receptor tyrosine kinases that have a unit on the outside that binds a ligand, usually a protein molecule, and then transmits that to the kinase domain on the inside that does signaling operations and does that transmission through a single helix. And on the face of it, this is a very puzzling kind of thing because what we know about these is typically that these units here are quite flexible. In other cases, like G protein-coupled receptors, we know there are many things that could happen in this transmission, and we know a lot, thanks to Chris and others here, but we also don't know the answers, we feel. I don't feel. And so we persist in trying to understand how this happens. And I'll talk to you quite a bit about histidine kinase receptors, for which, again, we face puzzles. In many of these cases, we face puzzles because we don't have enough structural information, though we might have other kinds of information. And there are many other kinds of receptors, including ones, I just have one example because we've done some work on Notch signaling, and these are even more complex in many cases, and I won't talk about them. So I'm going to talk about 4 kinds of receptors, and I'll try to blaze through these a little bit so that I don't take you too long a time. But I want to give you a little background on tyrosine kinase receptors, focusing on things that we ourselves have worked on to give you the idea. And the central idea that you'll know here, and a pivotal player in this is Josef Schlesinger in developing this notion, is the notion of ligand-mediated receptor dimerization. Here's a structure that we did some years ago on a molecule called stem cell factor, and we modeled a complex here based on the VEGF receptor, which is a homologue, if you will, of the KIT receptor that's shown here. But this picture I like because it gives you the idea in capsule form, I think, of what's really happening. So here we have a stromal cell that is presenting the stem cell factor. It also makes soluble versions, truncated, clipped off versions of this, that then activate the KIT receptor. And the KIT receptor would normally be floating around in the membrane of this stem cell that's going to be set into differentiation by the interaction with stem cell factor. Stem cell factor itself is a dimeric molecule, and that dimeric molecule, because of these associations, as we postulated here, would likely lead to the activation of this kinase unit in a way that I'll tell you about in a second. And a couple of years ago now, Schlesinger himself managed to get that entire complex. It's a beautiful accomplishment, and I'm very pleased because it really capsulizes what we thought was going on due to not just our own evidence, but evidence from a lot of other people at that time. So I think that really is the kind of idea. And the question of how it is that this then leads to the phosphorylation events that lead to activation, I think is importantly contributed to by work that Steve Hubbard did with me on the insulin receptor tyrosine kinase, which was in fact the first of these tyrosine kinase receptors. We have now many of these, dozens and dozens of these kinds of things. What we found in that structure was that there were 3 nearby tyrosine residues in this segment that's purple here. We call it the activation segment. And what comes to pass is that these are crucially important in the activation process. They get phosphorylated. And Steve later showed that that phosphorylation event, together with a peptide complex, binds in a certain specialized way. And my summary of all of this, it starts with his structure, which had a tyrosine residue blocking the place where we anticipated by correlation with the cyclic AMP kinase would be binding where the adenine moiety of the ATP would bind. So it's auto-inhibited. And that may happen in many cases, probably not a very general problem. But what I think is general is what's shown in the rest of this portfolio. Namely, that there needs to be some mechanism by which this activation loop can flip out. And when flipped out, it can get phosphorylated, as you see here, to make a phosphorylated unit. When flapped out, it can also alternatively come back in a conformation unlike that one that is a conformation that can bind ATP. If it can bind ATP, then this unit, to think of this as a pair on that stem cell factor, or on that KIT molecule, then this molecule can in principle be the enzyme to catalyze phosphorylation of this one where the tyrosine is accessible to phosphorylation. And the equilibrium is that when you phosphorylate this particular tyrosine, others could be phosphorylated as well by this molecule, but when you phosphorylate this, you tip the balance towards the ATP sensitive state, and ATP can bind, and now this is an activated kinase, and that is the activation process that we think is going on. The mediation by which this dimerization can happen happens in many ways, and I'm going to click through 3 or 4 of these, again referencing mostly our own work, but not exclusively. This is the fibroblast growth factor receptor, domains 2 and 3 of it, and the red is the FGF molecule itself. And this is a complex that we determined some years ago. And in this case, the individual FGF molecules are separate, but down this groove that's shown here in blue, heparin binds. Sulfated polysaccharides bind down this groove, and they, together with the FGF molecules, collaborate to become the dimer that then is the basis for activation of the kinases in this system. Another system that is exhibiting a wonderful set of controls and a very important system, the Egf receptor system, is illustrated here in 2 structures that were published a few years ago. This one from Dan Leahy, the first of the structures to be published, had the full external domain of one of the Egf receptors, here 3. And what you see is that it comprises 4 domains, 1, 2, 3, and 4. Comparatively, here they're called other names, L1, CR1, and L3. And what transpires here is that this hairpin, this beta hairpin, is blocked in this unligated state. The binding of the ligand, now there's it's a monomeric ligand. The ligand binds between L1 and L3, as shown here. This is TGF alpha, which is an analog of EGF, and it binds there. And when it does so, it pulls these 2 things together, pulls the domain 4 away, and when domain 4 is away, this finger matches up with the finger from a partner molecule. So what you see here in this yellow over maroon or whatever is the interaction that is responsible for dimerization in that case. So there are a lot of ways that this can happen. And another way that it can happen is I'll show you in an example from our work on CD4. What we know from CD4 is that CD4 is a molecule that is in the membrane, but it interacts cytoplasmically with a membrane-anchored Sark family member called LCK. LCK also interacts with CD8, which is another molecule we've worked on. And in the last of these slides, I just illustrate the point that in this case, the ligand is another cell. It's the MHC molecule on another cell. That activation process activates the T cell receptor. And what we think is responsible for that is a dimerization that happens in the CD4 molecule by virtue of this 4th domain of the CD4 molecule. The action head up here for interacting with MHC or with HIV is at this domain, but this domain is responsible for dimerization, and we actually measure that in solution as well. So for this part, I just want to call attention to these people, mainly Steve Hubbard and Debbie Stauber and Xuliang Zhang. Let me turn now to a really very active area of research for us right now, histidine kinase receptors. And these are primarily in prokaryotes, in virtually all through the bacteria and archaea, but also very heavily present in fungi and in many plants, but not at all in metazoan animals. So it's a kind of a puzzle as to why these were abandoned by metazoan animals, but that seems to be the case. And the system is what are called two-component systems. So there's a histidine kinase receptor or sensor protein that's shown here, and I'll say a little bit more about that, and also response regulator protein that is shown over to the right. And the process, the signaling process that we're wanting to explore is a signal binding to the sensor domain on the outside, or the periplasm of a bacterium oftentimes, and that then gets transmitted through what we believe to be a largely helical ensemble in the cell, and through a transmembrane segment that is minimally 4 helices, 2 from each of 2 domains, 2 protein subunits. And then the ATP bound to this histidine kinase unit phosphorylates a histidine here, and a phosphotransfer reaction from the histidine to an aspartic acid residue of this response regulator leads to conformational changes in the response regulator and activation processes. Activation processes typically are that this effector domain is typically a transcription factor. And I'll say a little more about some of those. So the histidine kinases, I'm not going to belabor, but they are very clearly identifiable by a number of named boxes here. The named boxes typically are all related to the contacts that are being made to the ATP molecule bound here, that is the agent for phosphotransfer. And we've worked on a few of these, and more importantly, arrived at a structure that is shown here, published a few years ago, of the entire cytoplasmic domain, we think, of an otherwise uncharacterized protein from Thermatoga maritima. So Alberto Marina in the laboratory did this work, and we did a number of studies on this that I won't take time to tell you about, but it led us to a number of hypotheses about how the reactions work that I did want to share with you. And it's the idea that this is the structure that we solved there, and that somehow or another the phosphotransfer reaction happens, leaving us now with, there's supposed to be a little yellow star here, as the phosphate group. And that then is the site to which the response regulator domain, the receiver domain of the response regulator can bind, as is illustrated here, leading us then to the phosphorylated response regulator that can go on to do actions. And then what this enzyme, what this histidine kinase receptor can also do is to act as a phosphatase and to promote the dephosphorylation of these phosphorylated molecules. So these molecules are both phosphatases and kinases, and in fact, our working idea is that there's no off. They're either dephosphorylating or they're phosphorylating, and that they don't really have, it's not a matter of activation so much as a decision of which way they go. And different histidine kinase receptors are favoring the phosphatase kind of activity as opposed to the kinase activity. Just recently, in fact tomorrow, Alberto has published this structure, which I'm very pleased to see, because the response regulator that he identified for this molecule, otherwise previously uncharacterized, turns out to bind precisely where we thought it would from other analogous studies. Let me turn to the sensor domains, which is something that we've been working on quite a lot for the last few years. And the way we've worked on it is the following manner. The reason that we've done this is that the sensor domains, it turned out, it was becoming apparent to me, were very diverse. So we did the following proteomic kind of an analysis. We realized that these proteins are characterized by 2 transmembrane segments, one that just is really at the end terminus of the protein, followed by the sensor domain, a transmembrane segment, and then the rest of this unit, including the histidine kinase. As I've told you, the histidine kinases have sequence signatures that are very identifiable, so we know a histidine kinase when we see one. We can easily identify transmembrane segments. We didn't think that we knew how to identify many sensor domains. So we said if we have these symbols, the transmembrane, and the kinase in it, and we have just 2 of these, there are others that have multiple ones, but we just focused on the 2s, and took all of those sequences that we knew at the time and divided them up into families. And the output of that analysis is illustrated here. We found in total 1,664 sequences that match that. Of these, that excludes a bunch down here that had fewer than 20 residues. That comes about because some of these sensor domains don't really exist. They have their little stubs here. There are molecules that are thermal sensors and molecules that interact, for example, with sensory rhodopsin that I'll show you in a minute, that are in that category. So they're important molecules, but they don't have these domains that we were trying to categorize. So we took these a couple thousand molecules, categorized them into families, and we found 500 families. Well, that doesn't sound like I made much progress, but more than half of them are singletons. There is no family, no other protein that has a good enough match for us to consider them within a family. But many others do match into families, and there are domains of different sizes in many of these, and so I'll show you that again in a little while. So what we ourselves and a few other people have done some of these, done other ones. These are ones that we've done. We've done lots of these kinds of proteins now. We have many of these examples. And we also have done an analysis comparing, in some cases, the structures with the sequences. And the thing that we've done with the sequences that I think is relatively unique is that we've found a way to map the distance between a pair of sequences into a position, a coordinate position in multidimensional space. And we can, so we basically do we do an eigenvalue problem on this. And here we just projected the first 2 eigenvalues and can separate these out. Now in this case, what we've done is to separate out the families done at a 10 to the minus 3 E-score way by blast type searches, and then separated them by the distances of family 51 to family 2, for example, all together, and all of these, and triangulating all of these by this analysis into the separate positions. And I think this is an interesting and useful categorization. But in addition, we can take, so family 1, 2, 3, 4 are all ones that are in the order of their population. So family 1 has the most members in the analysis, 2 the next, and so forth. So we've done structures I showed you on that previous slide of DCUS and DCTB, DuctB, and recently a couple of these others. So this is a case for family 1 where we've now, it's the same picture as that, but now this is spread out into subfamilies, so the different colored units are different subfamilies. And Zhen Zhang in the laboratory has done a sampling of structures. 5 of these are shown here from diverse sets of these families, and they still all are very similar, and we're beginning to try to understand what it is that differentiates them. So we've got a lot of information on these, and what we find is that they really are extremely diverse. They are all activating very similar histidine kinase molecules, but they are very diverse in their morphology outside. We have structures that are all alpha helical ones that I'll mainly stress, others that are like FOQ and DCUS and SID-A, PDC folds we call them. They're combinations of alphas and betas. There are others of these that are doubled up. DuctB is an example of that. And then we have doubled up all alpha structures, one that I'll spend quite a bit of time with you on. Others that look like periplasmic binding proteins, and we don't quite know how they work. And an all beta structure made out of beta propellers that we're still just beginning to understand. What really is critical here, I think, is dimerization, and it hasn't been easy, and let me just take a little bit of an aside on this. When we clip these molecules off, these domains off the membrane, they very often are forming dimers in our solutions, but more often than that, they're monomeric. And what we believe is that what we see in dimerization may often be representative of what is real, and that's what I'll try to emphasize to you. The affinities for dimerization may be very, very weak, but by theoretical arguments, if they were tethered to a membrane, as they are by the helices to which they are connected, that intrinsic affinity, as they are localized in a bilayer, is expected to be up by from 3 to 6 orders of magnitude, so many-fold higher affinities. In this case, this molecule behaved in our experiments in solution as a monomer, but in our crystals it formed a dimer, and the dimer looked like this. So we went together with Carrie Waldberger into a mutational analysis, and for example, we mutated these this glycine that was in juxtaposition, and also this charge pair, arginine and aspartic acid residue, in this study. And we have a beta-gal reporter assay that I won't take time to describe for you, but whereas the output, and this is a sensor that gives a signal when you have a very low concentration of magnesium in the medium, and it's believed to be a magnesium sensor in that way. So in the case of low magnesium, which is this bar here, the wild type is at these many units. And when you make the arginine to aspartic acid mutation, it's very depressed. When you make the aspartic to arginine mutation, it's very depressed. If you do them in combination, you recover a good bit of that activity. And the glycine mutation is also consistent with the idea that this is the right kind of idea for dimerization. There had been other models before that, but I think this is the right idea. And we've done many other examples of that since, and I'll show you a couple more in just a minute. But before I do that, let me get to the transmembrane part. The best idea that we have for the transmembrane part comes from this structure of a sensory rhodopsin with its transducer domain. So there's much more inside, nothing more outside. So this is one that is just a truncated, no sensor domain. But it's sensing the sensory rhodopsin state depending upon what its photoactive situation is, and that in some way modulates the activity of this receptor. For me, what it does is to provide me a model of what the transmembrane parts might look like. It could be that they're different in other cases, but in the case of the NAR-X molecule, together with nitrate, the ligand, each of these is somehow sensitive to some kind of ligand. This ligand is known, and it's nitrate or nitrite, and it binds at the interface between these two molecules in our crystal structures. And then by modeling, we can tack this onto this transmembrane segment that I showed you just a minute ago. And that, in turn, we think, as I'll show you in a second, would be connected up to other components on the inside of the cell. A very similar kind of arrangement can be made for this really radically different kind of structurally different molecule on the outside. It binds dicarboxylate, C4 dicarboxylates, in this case malate. It's DCUS is its name. And it's bound malate at these two sites that are way away from the interface, but yet the molecule is such that these pieces from the outside match up very nicely to the pieces that go across. So we think that this is how these molecules might work, but if you look at how that's connected up, and I won't spend time on this right now, but the orientation, so the way it goes through the membrane, according to the emanation of these ends, has to be different. So the wiring diagram that I showed for those two different pictures ends up being completely different, sort of opposite of one another. So that in turn is I think is an interesting finding from these studies. Well, how does the signal transmission go? And I've shown you some of the bits and pieces, so I'd like to try to put it together. What I'd really like to do to put it together is to solve the crystal structure of the entire thing, and that's what we have been striving to do, and not we alone, others have been trying to do it, and it has proved to be quite difficult. But because we have lots of little pieces together, we can make some kind of an idea of how they go. And some of these elements are in common and some are not. So this is the sensor domain. I've given you many examples of sensor domains. The transmembrane domain is going to be 4 or more helices from two molecules, as shown here. Here I've just reproduced the one that I showed you before from the sensory rhodopsin system. And then very often there's what's called a HAMP domain, but not always. It's like about a third of the cases for these molecules, a larger fraction of the chemotactic receptors that are cousins of these molecules. I won't spend time on that really. But this is a helix that comes down and then a loop back and another helix. And oftentimes there are other domains. In this case, there's a cis-rich domain that's on in the connection between the two for NARX, and then a dimerization unit here that has a helix hairpin kind of unit and the kinase domain. So that's the kind of scheme. And then the question, of course, is what happens here to cause something to change down here? And there are many things that have been discussed about this, and I'm going to just not try to go through the panoply of all these, but to give you some of the bits that seem to be from recent work operational. So we did the structure of the nitrate bound, I showed it to you, but we also did apo-NARX. It didn't form a dimer, and we didn't see it as a dimer, so we don't know really what it looks like as a dimer, but we can look conformationally at what the two things. They're quite a bit different, and we could superimpose them as shown here. And what transpires if you do the superposition is that the relative placements of the N and C terminal helices, which are the ones that are going to be going through the transmembrane part and on in, coming up from the N terminal domain along here, and then coming back, but ultimately inside the cell, the C terminal helices emanate as a unit. They're a twisted pair going through into the histidine kinase. So if you make a change of the relative positions of these, you have the chance to make a change on what's happening and could be perceived by the histidine kinase. And that, we think, is probably what's happening. And an interesting corroboration of that comes from the data that I illustrate to you here. So this is actually from Joe Falke in a recent review paper of our structures, actually, shows that when nitrate is bound to NARX, this C-terminal helix would be pulled up by this kind of a superposition, whereas the comparable thing that happens from work that Joe had worked out years ago on the aspartate receptor TAR from bacteria, which is a chemotactic receptor, but the outside domain part is a very similar kind of thing, leads to an oppositely directed transfer of the C-terminal helix relative to the N-terminal helix. And in this system, what happens is that when you add nitrate to the system, the output signal is a kinase signal. Phosphorylation events happen. You phosphorylate the histidine and onto the response regulator. In the case of TAR, if you add aspartates to the system, you activate the phosphatase. You dephosphorylate the system. Now, what was done by Manson and coworkers is to make a chimeric arrangement in which the outside part was the NARX domain, the sensor domain that I've just described to you, and the cytoplasmic part is the part that came from the TAR receptor. And in this case, when you add nitrate, instead of activating the kinase and activating the phosphatase, you activate the kinase. So it's oppositely directed, and it's completely consistent with the idea that the mechanism has something to do with this displacement of one helix relative to the other, and when you go in the opposite direction, you get an opposite output. So I want to spend a few minutes now with describing to you a system that we've been working on recently that I think illustrates many of these points and brings them together, and it's the TOR system. It's a system that responds to a molecule called trimethylamine N-oxide, and it does so in anaerobic metabolism to become the respiratory system. And the way it works is that TMAO, which I'll explain to you in a minute, trimethylamine oxide, binds to the TOR-T molecule, which is a periplasmic binding protein that associates with TOR-S, and I'll show you results of that. That result leads to an activation of this histidine here, and it turns out there's a phosphorelay system in between before it hits the response regulator, which when hit is dimerized, goes to the binding site for this operon of TOR-CAD, leads to the expression of TOR-C, which is a cytochrome molecule. Heme is incorporated in the periplasm, and TOR-A gets made with an assist from TOR-D. Then with the electrons coming to the reductase system, a molybdenum center, TMAO becomes the electron acceptor of that. It breaks TMAO into TMA, trimethylamine, which is a volatile compound. And if you've ever been around rotten fish, this is what you're smelling, TMA. So it's a very commonly occurring entity in sea life, basically, and important to many bacteria for their respiration in the absence of oxygen. Now, the TOR-S molecule is a structure that we reported on a month ago or so, and what its structure is, structurally, is that it has a 4-helix bundle down at the base here that's very much like the NARX and TAR molecules that I mentioned before, and that connects up to an upper domain with an additional 4-helix bundle, but this one now is left-handed, so it's right at the bottom, left at the top, and it's all twisted together. And we subsequently then did the complex of TOR-S with TOR-T. TOR-T is the periplasmic binding protein that is binding the ligand. So TOR-S doesn't get the ligand directly like NARX does or TAR does. It gets it indirectly by virtue of its associated partner protein. And the complex that we see is shown here. If we look down from above, what you see is these towers, if you will, of TOR-S and the TOR-T molecules on either side of this unit. So what the overall pattern of the TOR-S is that there's a tower bundle here, a 4-helix bundle. There's a pair of what we call distal bundles, these left-handed ones, a connector segment, and then the proximal bundles that are going into the membrane segment. So that then we ask the question really of what are the affinities of all of these? And the issue is a little bit complicated because we did measure the dissociation of the TOR-S from itself. We found that it had, for this particular molecule, which is Vibrio parahemolyticus, it's a 200-some micromolar affinity, so very weak affinity. So if you were to try to do analysis of this by analytical ultracentrifugation, which we were studying, it would be quite complex with all of these various equilibria that you would have to consider. So what we did instead was to make a fusion protein where we tied the C-terminal end of one molecule to the N-terminal end of a partner molecule and made that larger protein so that we now have this blocked together. So these are impossible, and we're now down to a simpler system that we could actually measure. We did the associations of that and found that these data were fitted best by far when we, well, I know by far, but they were fitted best. We actually had a hard time fitting it any other way by saying that there are that the binding events on the front side and the back side of this really are identical. So KD1 and KD2, these two binding events are the same. That was true in this case for the E. coli protein, which is a little, these are in the presence of TMAO, the affinity is pretty tight. In the apo state, it's a little looser. Getting confused. And more recently, what we have done is to, by isothermal titration calorimetry, to measure then for this same complex. Now we make the complex TOR-T onto this coupled TOR-S molecule and measure the binding of TMAO to that system, and we find that there are two distinct binding sites, one in the order of 1 micromolar, another 100 micromolar. So the overall system then, by this analysis, from a thermodynamic cycle analysis, is that we have binding energetics that have these two different states, and I'll show you in a minute that associates with our structural information. The binding that I illustrated here, this was modeled best when we had sequential binding. That is, this binding event had to happen before the next one. They're so disparate that it's a little difficult to make that statement unequivocally. That is, you could have asserted that this is a negative cooperativity, that it was the same to begin with, and that you only get it distinct because the first binding event decreases the affinity for the second. It may well be in this case that they are essentially at low affinity. The second site is low affinity, and I'll give you a reason for that in a second. So this affinity is in the same ballpark anyway of what you would calculate from the cycle for the TOR-T alone. this proved to be we haven't been able to do that experiment for technical reasons. So what is TOR-T? So TOR-T is a periplasmic binding protein, as I've said, and the closest relative to it that we can find in sequences is the ribose binding protein. And what you see here is a morphing between the bound and unbound state of ribose binding to ribose binding protein. In the case of TMAO binding to TOR-T, as it is associated with TOR-S, the TMAO, trimethylamine oxide, is bound into this site with these various residues associated to it, some of them binding to the oxygen, others making pi cation kinds of interactions, such as here and here. And all of these things are probably important to the activity of the molecule. And in fact, we have generated for this system a GAL reporter system for this that I show you a few results from here. The wild-type protein gives a response to TMAO that's at this level. If you make mutations, for example, making this an asparagine, this aspartic acid, or this tyrosine into a leucine, you abolish mostly the reaction. It turned out that this, surprise to us, didn't make a change. But again, this getting rid of the oxygen of that into otherwise a very conservative mutation is deleterious. Now, if you look at the structure as a whole, what you find is that the binding of TOR-T, or not having it, the apo structure is very similar. Superficially, it's very hard for you to tell the difference. But in both cases, the TOR-T molecules are in essentially the closed state. So in this system, that clamshell opening and closing doesn't seem to be happening in the operational state, which is with the association to TOR-S. And that happens because there's a C-terminal extension here, not present in ribose binding protein, this red part that is holding all of that together. But the apo structure is quite asymmetric, distinctly asymmetric, although, as you can see superficially, not very similar, not very dissimilar. There is a difference in the atomic mobilities measured by our crystallographic B factors, and you can see from the redness and yellowness here, as opposed to the blues and greens, that the apo state is more flexible at the region where the TOR-T is bound. Now, if we look in detail at these complexes, what we see is a superposition here of one of these molecules, TOR-T double prime we call it, shown in gray, onto the one where the C-terminal domain is colored blue, the N-terminal domain is colored yellow for the TMAO-bound state. And the same coloring scheme is over here, but now it's the TOR-T prime. What differentiates these two two things differentiate them. One is that in this case, there's a loop that is, well, in this case, in TOR-T prime, there's a loop that's disordered right in this vicinity that is ordered back there. And in this blow-up, you can see that better. The ordered loop here is plugged in, and when it does so, it sort of prizes this open from what it would be in the TMAO state by a couple of degrees, and when it's disordered, it's closed down. So these two things are associated with other changes that happen. But first of all, let me say that this loop itself is functionally important. That is, if you mutate this glycine residue right here into an alanine, you essentially kill the activity. Maybe the protein isn't even viable since it's so dead. But if you mutate one or the other of these, you cause a shift in the binding for TMAO. But if you mutate both of them, they're basically it shifted a few orders of magnitude in that instance. So this loop needs to be flexible. Glycine to alanine mutations are going to make these flexible. I should also have said these two out of these three glycines are in conformations that only glycine is allowed to have. So what are the consequences of those interactions? Well, the first thing that we observe when we do a comparison, a detailed comparison of the apo state to the TMAO state, is that the tower domains are virtually identical. So what you see here is the tower domains superimposed, but when you have those superimposed, the bottom parts are not any longer well superimposed. They can be brought into better superposition by a rotation, a lateral swing about these axes that are shown here, about 3 degrees in the same direction. And now you see these are, not only are these superimposed, these units are superimposed, these units are superimposed. And now further, if you take these positions and from this unit ask what does it take to superimpose this, superimposing these distal domains, how do we get the proximal domain superimposed? We do that by further swings, but now these are about axes that instead of being lateral are along the axis of each of these respective units, and their swings by about 3 and 8 degrees respectively. All of that correlates with what's happening to TOR-T. If you look down the axis at the TOR-T molecules, T double prime, the entrance loop engaged, this one flexed out, what you see is that the TOR-Ts didn't just move on their own in a similar way, partly opening, a little bit opening. They move toward one another. You see, they both are going like this in this complicated way. And if you look at the difference between these two, the difference in the N-terminal domains is about 6 degrees. Countering that, interestingly, the proximal domain, the membrane-associated domain, is twisted that 8.4 degrees and 3.4 degrees that I showed you. Those twists are going in the opposite direction. So what you do at the top is counterbalanced by what you do at the bottom. How this plays into the signaling is not so clear, except to say that, again, a bit like I showed you for NARX and for TAR, if we do the superpositions as indicated over there, what we find is that there's a relative displacement of the N and C terminal helices again. In this case, it's pretty subtle, about a half an angstrom unit, but it may be something that's responsible for the signaling basis. And again, the same kind of idea that we'll signal, in this case, through this larger unit on the outside and through entities on to the inside. So that's my story about the histidine kinase receptors, and I want to give you a quick sneak at GPCRs and cis loop receptors in the remaining moments, but let me credit particularly the work on TOR-S for Jason Moore and generally on Jonah Chung, and I've mentioned some others along the way. So G-protein coupled receptors, I'm not going to say very much. I just do want to get a couple of points across. So there are many kinds of G-protein coupled receptors, as you will know. A very common thing. This is a picture of rhodopsin, thanks to the work from Chris and others. And we've been doing a lot of work on serotonin receptors. I'll show you just one little bit of that. And on follicle-stimulating hormone receptors. Now, I wanted to make the point here that these have very large outside domains, and these have almost no parts that are on the outside of the cell. But, and I'll show you just this little bit of the structure that we did of the hormone binding portion from FSH receptor with FSH. FSH itself is 2 domains and 2 proteins, an alpha protein and a beta protein. The alpha is in green here. And I won't go through the arguments for you, but we think that this is the part that's near to the membrane. And the picture that I have reinforces that. And the puzzle that I want to leave you with is that many of the biogenic amine receptors, certain serotonin receptors, epinephrine beta receptors, bind their ligands in a pocket that's very similar to the placement of retinal in rhodopsin, and they activate G-alpha S, which can stimulate adenylyl cyclase, for example. And the FSH receptor does the very same thing. The bottom line is that it stimulates G-alpha S and activates cyclic AMP-dependent cyclic AMP production through adenylyl cyclase. But it does so despite the fact that it doesn't have, it's not a small ligand, it's a huge protein that's binding to the outside, and it interacts. So my thought, and this is just a hunch, but there are plenty of cases that we do know about where peptides insert themselves into the placement where the ligand would normally bind. And it's a possibility that these tips, which are conservative, might be doing that. And what we know when we superimpose the structures of FSH molecules that are bound to the to the FSH in green onto the structure that was done with it free, what happens is that the free state goes from, it's all the same out here, but these tips then move from where they were free to where they are complex. Here's another view of it in the other direction. And our thinking is that that interaction ultimately stimulates the activity of the protein. Now, the molecule that we solved is a dimeric structure, and there's a lot of evidence from other people's work, some papers here that I think are nice pieces of evidence of complementation, molecules that I schematize here, in which one molecule is made with a defective hormone binding unit, and another molecule is made with an effective hormone binding unit, this pinkish color here, but with a defective transmembrane region. So this molecule can bind, this molecule cannot signal, this molecule cannot bind, this molecule cannot signal. If you transfect either one alone, you get no signal out. If you transfect them together, they complement and you get a signal out. So that has been a puzzle to us in the sense that we, if you took on face value that dimer that we had produced was the right thing, it would be very difficult to see how we can make the activation of this to happen. If we, on the other hand, have sort of a different kind of an asymmetric unit, maybe that can work. And one piece of evidence that this may be true is that from a different system, from a relative, TSHR, that is the case. There's what's been described as negative cooperativity. There's one 0.2 nanomolar or 200 picomolar binding affinity and another that has 20 nanomolar. So just a little bit on serotonin receptors, and this is it really. We have done a number of mutational studies on serotonin receptors that include making mutations that are a bit analogous to what I've told you about. This one, first of all, D135N is a mutation that is known to decrease the binding affinity for the ligand serotonin, and another Y369F is one that causes a defect in the signaling of the molecule. The binding is actually potentiated. And in our experiments, if we look for IP production, this is a G-alpha Q system, you basically kill the activity with either of these two mutations. But if we co-transfect the two mutated proteins, we get a robust signaling out, suggesting that somehow or another these two molecules are talking to one another. The easiest model is one in which they are dimerized in some fashion, such as this. And a favorite model of many people is that somehow or another one heterotrimeric G protein might bind to such a dimer. And that work, I should have said, is I credit particularly Ching Fan on the FSH receptor work and Filippo Mancia on the very abbreviated story on serotonin receptor that I've given you. And let me now just very quickly race through some work that I think is really quite interesting on cis-loop ion channel receptors. These molecules are acetylcholine receptors for short. There are other kinds. Some of them bind serotonin, for example, and other glycine, other things. And they all have 5 subunits, and they all have a cis-loop. We'll see, I think, maybe a picture of the cis-loop. And they have this ligand binding domain on the outside, 4 transmembrane helices that go through and end up making in the membrane a pore. So there are some structural precedents for what I want to tell you about. The first is that the first important information on this system really came from a protein that is known as acetylcholine binding protein. It doesn't have a transmembrane segment. It's thought to somehow be involved in regulating acetylcholine for the receptors, but it led us to the first good picture of what these molecules would be like from work of Tisha Sixma that is shown here, and others have done work on this. And then work by cryo-electron microscopy done by Nigel Unwin that very importantly has worked on the muscle protein, giving a picture of the whole molecule. It's at somewhat limited resolution, 4 angstroms, but to my eye, a relatively lame 4 angstroms in the sense that the helices are just tubes. It's very hard to get very much definition of what's going on, but it gives us an idea of what's going on and good enough to get a rough model. And the green part here is probably a pretty secure bases because it's modeled on the acetylcholine receptor binding protein. So what we have done work on that I want to briefly skirt through is work on an acetylcholine receptor that's called alpha-9. The whole family in humans is shown here. There are many of them. Alpha-9 and 10 are involved in the efferent auditory system as indicated here, their feedback system basically. And alpha-9 can form channels. We actually think that the functional channels are probably alpha-9, alpha-10 heteropentamers, but alpha-9 can form heteropentamers, and we had hoped that we might be able to form those by making them. We didn't. We ended up getting what were essentially monomeric proteins, and I think nevertheless that we learned quite a bit by doing, if you will, a little pharmacology, that we did bindings to many different compounds, including, importantly, that I'll tell you about, carbamylcholine, an agonist that's a sort of a non-hydrolyzable or weakly hydrolyzable version of acetylcholine, and on this robust antagonist, methyl-licoconitine. So the structures are, the molecules are shown here. One of them is a small protein, conotoxin. But the ones that I'm going to tell you about are these two, and they bound in these sites like that. The two structures, the structures all look very similar to one another and somewhat distinctly different, but also by and large similar to the acetylcholine binding protein. Where they really differ is down here at the cis loop that's diagnostic of these, characteristic of these proteins. And then loop C is the place where the ligand actually binds. And the cis loop has this particular conformation that's shown here that we see for the first time, and I think is legitimate conformation. So the agonist binding leads to that loop coming through a very distinctive conformational change that you see here. This is the morphing from the alpha-9 protein from the MLA antagonist state down to the agonist state. And the very same motions, very, very, very similar motions happen with the binding of, same binding event in the case of the isolated binding protein. Now, if you look at the whole molecule, though, there's a very different transmission of that binding event into the protein. In the case of the legitimate receptor protein, the binding event here leads to a swaying of the entire molecule, whereas the binding in the acetylcholine binding protein is localized just to this binding loop. It doesn't get transmitted into the rest of the protein, even though the proteins are very similar to one another. And we think that the reason for that has to do with the nature of the interactions in the molecule. Now, of course, the binding protein doesn't have to transmit any signals, and it doesn't. And it has, what you see here are two staves out of the beta barrel, out of the five-stranded, out of the five subunit acetylcholine binding protein, with the neighboring molecules imprinted onto those in these red footprints. When we make a homology model, superimposing our alpha-9 structure onto this is the footprint. Basically, there's little footprint down below. There are holes in between these things. This is very flexible down below. And what we think is happening is illustrated here. Namely, that if we superimpose the parts that are shown in red, that is one isolated subdomain that we can distinctly show by a computational method, and make this closing, what happens is that the bottom part swings. The cis loop is swinging. So the top is isolated by this pentameric arrangement, and the bottom units can swing independently of that in this modeling. And we think this is really transmitted through the junction to the transmembrane domain via the cis loop unit poking into the hydrophobic content among the four helices, in between the four helices, contacting this very important loop here, and a lot of mutational studies to support this that I'm sloughing over. So the bottom line is this. Here's the take-home message from this section. So if we take this model that's shown here, and we have and analyze the swing that I showed you in that automation, there's a pivot around a point that's like this. And what I've illustrated are two of the subunits out of a pentamer of this kind. If we do a calculation using an elastic network model that we've developed. For the transmembrane part, using a homology model that we've built into the Nigel Unwin structure, we turn out to have a pivot point that's about here. I've illustrated it at this point for those domains. And they're coupled by this juncture that I showed you before. Now, the analysis by 3 different analyses, the first of them being by my colleague Arthur Carlin, about where is the gate, where is the block in these acetylcholine receptors. All 3 of these analyses illustrated here say it's down here at the base. Down at this point is where there's the constriction that is limiting the passage of ions. So what the receptor has to do in response to a ligand is open that gate. And our idea is this, that as we swing from the closed-like state with MLA to the acetylcholine state, modeled from our carbamylcholine, we go from the blue to the red. As we do that, we push on these molecules down here at the juncture, and all it can do to respond is to rotate about this axis that I've shown you here. So moving up above here pushes on that, which is going in the direction of opening the gate. And I'll just close by showing you the automation of that. First of all, from the side, you see how this kind of motion is happening. Here's the binding loop coming down on the ligand there and there. And here's the cis loops moving in response to that. You can see it a little better in this case. So we move in there, and that's opening down below. And if we look from the top view, you see that happening in this fashion. Now, this is largely conjecture. We haven't got these structures. We would love to have these structures, but I think this is a model that might likely hold to the test of time and additional experiments. And this is the work of a former student, Ruiz Xu. So with that, I believe I'm at the end, and I thank you very much for your perseverance. I'm sorry I'm a couple minutes over, but it's been a pleasure talking.
Question more on philosophy or your comments, how you see this domain structure, that you're trying to understand this movement throughout from the binding side to the intracellular domain.
Right.
How much you think those models will need to be in the future modified? Of course, you don't. This is your guess, so forgive me for just. I wanted just to hear your opinion on that. How much this will be modified as we get the structure of entire molecules? The last model, what you showed, the transmembrane helices will be interacting with lipids, will be constrained by the lipid interaction. So, how many of that prediction of movements from soluble domain is going to hold true in the future?
So I think that's, so the answer is really hard to say, of course. I think lipids are going to be very important for a lot of these systems, but I do think that if you take this last example of the acetylcholine receptor, I said I didn't need water, but maybe I do. That molecule is pretty, there are a lot of interactions between the individual protein molecules in that. So I think there's a lot to hold that together. And functionally, it seems to me that it has to be not so far different from the picture that the EM has given. It's a poor resolution and all that, but I don't think has much possibility to be radically different or radically modified by the lipids. But when you come to some of the other cases where there are many more pieces in the structure, then I think many fewer pieces in the structure, then I think there's much more possibility that it could be rearranged on the basis of other interactions with the lipids or other components. But and in fact, here's a place where I differ with Nigel Unwin's analysis. He made an interpretation that had the helix, the pore lining helices moving relative to the others. Our computations say that's not going to happen. And I think a lot of common sense from structural biologists' point of view would say that the integrity of that 4-helix bundle is going to be
very, very important to the interactions of everything and the interactions of neighbors. So I guess I'm fairly optimistic about things like that, but there are going to be, in cases like I was telling you about with the TOR-S system, or I think histidine kinases in general, I think that the signaling reactions that are happening are based on pretty subtle changes. When we're talking about half angstrom changes or something, or 1 angstrom changes in the relative positions of helices, it's believable to me that that's enough to do the job. And particularly when you can't have something swinging radically if it's got to be active in both states, right? Whatever you do, it's got to be active. But what we really are often seeing are, I think, in many of our crystallographic analyses, is things that are more symmetric than is real. So a lot of what we've seen are symmetric molecules. Our analysis of this situation, so what I showed you is that the TMAO state of TOR-S is symmetric, the APO state is asymmetric. In the case of the aspartate receptor, it's clear from the crystallography that half of the sites are occupied, aspartate on one molecule, not on the other. But in the absence of aspartate, the molecule is symmetric. So I think this idea of symmetry versus asymmetry is going to be crucial. And in some of the cases where we've seen something symmetric in our crystal structures, maybe it's not really symmetric, that the active or the important molecule, important state will be changed from that. In that case, I think maybe it's going to be a subtle change. In other cases, of course, we have to be open to the idea that what we're guessing at the moment may well be quite off the mark.
So let me have one more very quick question to your first part of your presentation on tyrosine kinases. You mentioned that you favored this model of dimerization induced by ligand, but the extracellular domain has its own affinity to each other on millimolar. If you extend it to the plane of membranes, that may be nanomolar affinity without any ligand. So is that consistent? Is it really true?
When you say that, I'm not sure. I agree with you about the millimolar to micromolar or more, if that's the observation. I don't know which system. Which one?
Slide on the tyrosine kinase. I don't
Oh no. Okay. So that one. Yeah. Okay. That's a good. That was shown. We did that measurement on CD4. So the CD4 one, that's millimolar. Now, I learned about this from Henry Metzger, who had done a calculation with Alan Minton on this, which I cite many times, love it. But I learned it when I told him about this result. And I had done my own calculation, which I think is probably right. There are something like 10 to the 4 CD4 molecules on a T cell. A T cell is 10 microns across, and if it sticks up 100 angstroms, you do the calculation, you find out that if it's evenly distributed around the cell, that means that the local concentration of these CD4 molecules is on the order of micromolar. But if you then, as we know from things, for example, from the idea of the immunosome or what is it called, the immune synapse, I think Mark Davis calls it, that these molecules, when they interact with their target molecules, when the T cell interacts with the antigen-presenting cell, a bunch of molecules migrate around the surface and congregate together, including in that synapse, this immune synapse, CD4. So there's a concentration mechanism. So the critical thing is that you have to have, for whatever the system, you have to have the signal be sensitive to, you have to have the concentrations be the right ones for the affinities that are there. So I think that the correct affinity of CD4 for its partner CD4 molecule on the surface of the cell, probably in however you normalize for the membrane, is going to be in the micromolar level, and we exceed that. The system is stable because it's not signaling in the absence of the partner cell, the antigen-presenting cell. But this is a valid question, and it's an extremely difficult thing to study, because to get something equivalent to the binding constant for something that's tethered to a membrane is it's hard to even describe in an analytical way.
I have a question. In that last part of your talk, the open and the closed stage, didn't look like there was much movement.
Not much, right?
So, is there any FRET evidence to show that what you're seeing there is about the right kind of distances?
Well, I mean, you can do calculations about what the the hole size is, and it's particularly it probably is a very sensitive number. We don't really know, and we don't have accurate enough models to do it from that point of view, I guess. What I think is encouraging is that it's making the what becomes open, you know, other things become closed, but what becomes open is the part where it is known experimentally the blockages to be, and how severe the blockages to be. Clearly, the blockages have to be in some way. I mean, you could imagine something where you're swinging a group into a block and you swing it out, and there's a possibility, I suppose, that some kind of changes of that kind are happening. But here's an alternative model that I think that fits. What we do need to do, what you have to understand, of course, is that the binding event way out there someplace is going to have to open that hole. And so there's got to, and it's presumably a concerted operation because the 5 units need to be involved in some fashion, or in many of the cases, 2 out of the 5. So I don't have a good answer for it. We need many, many kinds of experiments to get to the bottom of these, of course.
But that difference would have to be bigger than protein breathing, right?
Well, it's some, yeah, to be, well, I mean, obviously anything is going to leak at some level. But, right, so the idea is that the ligand binding, which has its own energetics from that interaction, has to be sufficient to make all of these motions, and that ought to be tipping the balance way beyond what the KT levels would be versus what a meaningful flow of ions would be for downstream processes. So, of course, the system needs to be secure against inadvertent openings.
Any other questions?
Do you have any general ideas about themes that might either be common or uncommon between transporters and transmembrane signalers? And one thing that kind of struck me from your talk is in a lot of transporters, the molecule will be a pseudo-dimer, you know, a gene fusion or something like that, where the second half is a repeat of the first half or something along those lines. But it seems like you don't see that very much with signaling proteins. The other thing I thought was that overall, it almost seems like conformational changes in signaling proteins are bigger than what you actually see with transmembrane proteins that are shuttling a molecule from one side to the other. And
well, I think that depends on some of the transporter. I mean, the working idea for the transporters. Many of the transporters are things that are many transporters are thought to flip between 2 states, right? And an outside occluded to an inside occluded to maybe some intermediate state. And those conformational changes, which haven't been properly seen within the same molecule, I believe, to this point, have been documented by homologous molecules for one state or the other, and we believe a modeling that would say it would go flipping and flipping. Same is basically true for the ABC transporters. So I think I think the conformational changes in those cases are easily on the same scale as the ones I've been telling about today, particularly the ones that we're struggling to actually define for the histidine kinase receptors. So I'm not sure that that's really true. But when it comes to channels, many of the channels can be very subtle. And maybe that's what I'm trying to say to Chris on the cis loop channels, that they tend to be, you know, once you open the hole enough, then the ions can go down their gradients. That's all that's needed. They're not actively transporting, but you do have to control the pore. So some of the channels, I think, are very subtle in how they move.
One last question? No. Okay, let's